It's really nice to have Alejandro Couce today giving the seminar. So Alejandro did his PhD in Madrid with Jesus Vázquez, and then he moved to Paris, where he did a postdoc in the group of Olivier Tenaillon, and then moved northern again to London in Imperial College, and he's now in Madrid for the last 2 years, and has started his own group on a tenure track position. And so I think today will give us some overview on his work on the role of mutation in evolution. Thank you, Alejandro, for accepting the invitation, and we're listening to you.
Okay, thank you very much. Let me just set everything smoothly. So I guess that you all are watching my first slide, right?
Yes.
So I was saying, thank you very much, all the organizing committee for having me, especially Stephanie, who was the one who actually handled me on a one-to-one basis. And to me, it's a great privilege to speak in front of this audience and also, you know, talk about this topic, because I'm fascinated by the role of bias in mutation in evolution. Is something that I sort of have been a little bit obsessed since my PhD. And so I'm going to share with you a little overview of my work over the years and try to make something coherent out of it. And so, without further ado, I'm going to start right away. You see, if you haven't read the abstract for my talk, I'm going to be talking about the evolutionary consequences of biased mutation rates. And I'm going to talk specifically about 3 consequences. Okay, the first one will be the role of mutation bias on directed adaptation. And then I'm going to be talking about its role in genome evolution, and lastly on the evolution of the mutation rate itself. So I'm going to start right away with this cartoon, which I put it here just to illustrate this idea that we often have in evolution about thinking on the evolutionary process as navigating different adaptive paths. Okay, and this navigation among those paths is driven by selection. In this particular cartoon, we will have to discuss which has more fitness, if the fountain of eternal youth or the fountain of bacon. And this we're going to be talking about this choice of different pathways. And I think that the first time in which this concept of thinking of evolution on navigating pathways goes back to the seminal work by Wright, although I will be very happy to be corrected on that, but surely is one of the first names to put this concept on top of the table. And so for those of you that maybe are not so familiar with that, with what Wright was trying to depict here, was the idea of you start with a wild type, which will be this plus sign here, and say that you have 4 available beneficial mutations, 5 beneficial mutations, A, B, C, D, or E, could be alleles probably. And the idea is that you can have all these 5 different paths, but then you consider the combination of double mutants, of double alleles, then the number of paths starts to multiply, and you can repeat the same process for triple mutants and quadruple mutants until you reach what could be, for instance, the optimal genotype that has these 5 mutations. And so this is something that, this type of concept, I was first exposed to that when I was an undergrad, and it really got my imagination. I think it's super cool, this kind of way of thinking about evolution. And so when I was starting my PhD in Madrid, this paper by Dan Van Rijk, that I understand that was a speaker recently in this very same series of seminar, this paper by Dan came along, and it really blew my mind. I guess to some other people too, because it's a highly influential paper. So essentially what they were doing is try to mimic, have a tractable experimental system in which you can sort of do what Wright was imagining here in 1932. Okay, so now it's 2006, and they got this enzyme that confers resistance to antibiotic, which is called the 10-beta-lactamase, for those of you that are into these kind of things. And essentially they knew this is a very well-known enzyme because it has importance in clinical settings, So they knew about 5 different mutations, and they essentially constructed through, site-directed mutagenesis, all the different single mutants, double mutants, triple mutants, and so on. So exactly what Wright was imagining. And so mind you that this network that we are seeing here in this slide is just a subnetwork. So it's just the subset of trajectories that they found that could be navigated by natural selection. What I mean by that is that each step in those graphs imply an increased fitness. Okay, fitness here was measured simply as antibiotic resistance, which is those little numbers that you see in each one of those circles. Okay, so you go from the wild type that had a, forget about the units, but it's a number that is 0.09, and it goes all the way up to something that is 4,100. And so they have all this information, and they apply a very simple population genetics model, and they came up with this with those nice arrows that are showing you which pathways are the most probable to be, throttled by populations, simply guided by selection. And well, I really love this picture. I think it is clear by now that I really love this one. And so I was a young PhD student, and I was super excited with this thing, and got my my imagination flying. And then they say, well, all in this population genetics model is based on the only sort of the directional force here is natural selection. Okay, that's something that was already present on Wright's depiction, but it's here super clear, because the only parameter that will dictate the direction and the thickness of those arrows is how much change in antibiotic resistance you will going from here to here, right? So this is the prediction based solely in natural selection, okay, and increasing adaptation. But of course, as you might have guessed, what would happen if the different propensity with which different mutations could happen, it's also a variable that you can consider. And this is, I would say that this is something that is very natural to think in this type of model system, because we know very clearly which are the identity of the mutations that they were introducing here. I mean, at the level of nucleotide. And so it's very natural to think, well, we know that certain nucleotide substitutions could be, could happen with a higher probability than others. So what would happen if you add to this sort of depiction the idea that some mutational changes could be much more likely than others? Okay, so the idea would be simply like, well, could you just, I don't know, push the whole system and make the populations to adapt through, say, suboptimal paths? So I mean, at the end, they will probably reach the same, the best combination of mutations, but maybe instead of doing, statistically speaking, following the most likely one according to their calculations, maybe if you have a very strong mutational bias operating, you could push these populations through suboptimal paths. And who knows, you can start imagining, and maybe if you have high enough mutational biases, maybe you can even bump into some other branch here that has not been explored in their experiment, and you could reach perhaps another completely different solution. So those are the kind of ideas that I was having back then, and I think they are quite intuitive. But as usually happens with intuitive ideas, well, sometimes if you think carefully about them, they are not so trivial anymore. So let me just do a consideration about this proposition that I just said about the impact that mutational biases could have on the navigability of this kind of a network of pathways. And so there's a first assumption that we are doing here, and that is that we are not in this kind of a scenario. So what I'm depicting here is this idea of the adaptive landscape. You have the surface that will be, displaying would be the space of genotypes, and the height will be fitness. And so imagine that we start with this population, and we have a scenario in which you have a very strong mutational bias that is, driving the population towards one direction in genotypic space. And you have this other case in which is driving populations towards another direction. Of course, if this adaptive landscape have this sort of shape, is smooth, then it shouldn't really matter having, mutational biases, because at the end, you will reach the same point, right? So when we are saying that mutational biases could have an effect, maybe we are assuming implicitly that we are talking about what people call as rugged adaptive landscapes, in which you have multiple isolated, suboptimal peaks, and you can have one or more global peaks. And so in this kind of scenario, intuitively, you can think, well, maybe if you have a population that has certain tendency to go through this path, it might get stuck in this particular, local peak, okay? And again, this is just the first approximation to the problem, but then you can think, well, there's more to it than just this discussion, because what would happen if, in the case of a rugged fitness landscape, you have a very large population size, which is typically the case when we are working with bacteria. We do experiment with bacteria. Typical population size, even in small microtiter plates, could be 10 to the 7, so 10 million cells per milliliter. And so the idea is that even in this scenario, if you have a very large population, you're going to have possibly all types of mutants going on, regardless if you have a certain tendency to generate more often one type or another. But at the end of the day, the populations will be able to explore, at least the most accessible pathways. And so it doesn't really matter if you have a strong mutational bias, because at the end, the natural selection will completely do its job perfectly, and all the populations will end up at the same sort of optimal solution. And so that's sort of the theoretical conceptual considerations that I wanted to share with you. And of course, what we did was to try to test that experimentally, okay? So the first thing that I have to say before I move on to explain actually the first paper of this series is to explain a little bit what I mean by mutation biases, and especially in which type of scenarios we will expect to have strong mutational biases, okay? And so, as you know, mutations are inevitable, and they can happen for many different sources, from the intrinsic sort of infidelity of the replication machinery that is copying genomes, to all the different physicochemical agents that are extrinsic to cells, but also intrinsic to cells that are, insulting DNA. And of course, cells have evolved a variety of systems that will prevent and repair this type of DNA damage, and they will therefore prevent mutations. And so when you, in the laboratory, you can make a knockout of one of these pathways, and then, of course, you get what is called a mutator bacteria. We are going to be talking a lot about mutators or hypermutators, depending on how do you want to call them. But essentially, there are mutants that lack one of those systems. And as a consequence, they have a highly elevated mutation rate. And I'm going to illustrate that with just a very simple experiment. This is a loan of E. coli cells, okay, in which I just put a couple of droplets of antibiotics. Never mind the names, but this is one antibiotic and this is another antibiotic. And what you see here is the halo of inhibition, okay, of this drug. Okay, so this is, to be absolutely clear, this dense opaque thing here is cells growing, and in this clear sort of circle, there's no growth, okay? But you see little colonies, okay, in some parts, and those are, of course, resistant mutants. And this is what a wild type E. coli cell look like in terms of, how often it will produce resistant mutants. And that's what it looks like if you use one of those hypermutator variants that are lacking one of the particular systems that I was discussing before. You see that the picture changes a lot. Now you have a lot of resistant mutants growing inside those halos, okay? And as you can imagine, mutators are of interest not only to evolutionists or geneticists, but they have an importance in clinical settings because they are very good at adapting to anything. And as you can see here, to antibiotics. as a matter of fact, here I'm sharing with you results from a screening that we did on urinary tract infection isolates. And you see each one of these panels is a different patient. And we detected in 4 of the patients on this cohort of 80 that they had strong mutators going on. So it's a 5%. There's other types of infections in which the prevalence is even higher. It could reach even 40, 50% in cystic fibrosis patients. But the idea is that mutators are a real thing in the natural world, okay? And what is very interesting about mutators is that they do not only elevate mutation rate, generally, but instead they are very picky about which type of mutations they elevate. So you see here, I'm just sharing the header of a very famous table from this paper, in which they are simply listing different genes and what is the sort of mutational specificity. In our language will be the type of mutational biases that they will introduce. And so you see that a particular gene called MUT-C will generate preferentially transversions. Another one called MUT-Y will generate other type of transversions. And all these 3 genes here will generate, transitions and frame shifts. So this sort of idiosyncrasy, it's what's going to be very interesting to us now, because those type of variants will create opportunity to peek into what would happen if you elevate specific type of mutations, if you introduce strong mutational biases. And so just one more thing before we start discussing the other paper about mutators and mutational biases is that, as you can imagine, in some cases, specifically when adaptation depends on very few sites, it can happen that simply by favoring changes from A to T or changes from G to C, you're just hitting the right spot, or you are hitting the bad spots. And so those are data from a paper that we will discuss later in the talk, in which we will just isolating single-step mutants that are conferring resistance to antibiotics. In this case, it's streptomycin. And I'm simply just showing you the different aspect they have. So all of them are single point mutants in the same gene that are conferring resistance to the same antibiotic. And you see here that, for instance, this MUT-T mutator is generating big fat colonies, while this MUT-Y is generating very small ones. And if you can check another antibiotic and then, the tables are reversed. Now it looks like it's the MUT-Y who tends to generate the big fat colonies, although not that fat as those ones, but are generally, bigger. They are growing better than the ones generated by the other mutator. So this type of idiosyncrasy, of course, simply boils down to what specific type of change you've made on which specific residue. Okay, we're going to discuss about that later, but this is just to sort of give you a justification of, those mutational biases are something that are real and are very, easy observable in the laboratory and also in nature. Okay, so going back to the first idea. Okay, we have Van Rijk's system, which is this beta-lactamase, this enzyme that confers resistance to one antibiotic, and they have these different 5 mutations that, when combined, will generate a super highly fit enzyme. And we want to check what will happen if you sort of navigate through this network, having the mutational bias switch on, okay, and try to see which type of a scenario we are. In particular, if, with the typical population size that we see in bacteria, something like this extreme case could really happen. I mean, this is the hypothesis that we want to test. So one thing that is very, very interesting about Van Rijk's system is that if you look at all the different mutations that they can happen, most of them are transitions. Okay, so most of them are just this type of change, GC to AT or AT to GC. Okay, and these type of changes are happen to be the speciality of one type of mutators, what are called the mismatch repair mutators, which you can get this type of bias just by deleting one of these 3 genes. Okay, So our idea was that maybe we can just repeat sort of Dan Van Rijk's experiment, taking this antibiotic-resistant enzyme and evolve it towards the same, to increase the resistance to a new antibiotic. But we can do it in conditions in which transitions are the predominant type of mutations that it happen. And we can compare that to a condition in which transversions are the dominant type of mutations that would happen, and see if there's some difference, okay? And so we follow a classical experimental evolution protocol. We have our two different mutators and also a wild type as a control. And we were doing serial passaging, and we were increasing the concentration of the antibiotic every other day, okay? And so this is sort of the general outcome of the experiment. You see here in the x-axis, we have the concentration of antibiotic that is doubling every second day. And we started with 96 parallel populations. And as you would expect, at the beginning, they all fare well. But as soon as, the antibiotic starts to be at high concentration, they start to become extinct. The first one will be the wild type, which is, something expected because its mutation rate is much smaller. But you see that despite the fact that our prediction was that one of these two mutators, particularly the red one, is the mutator that is generating predominantly transitions, so it should be super good, should go super fast across this network that we were depicting before. We don't see much differences with when you compare that to the other mutator, which is just doing transversions, and there's no transversion that we are aware of that could really make something usable in this enzyme. Okay, so that's the overall result. But the real, the interesting stuff comes when you look at the sequence, right? So first we sequenced all the different antibiotic-resistant enzymes, and what we see here are 5 mutations, of which 4 are present in Dan Van Rijk's study. And what you see here, the colors is telling you, well, the red will be this transition-prone mutator, the blue one will be this transversion-prone mutator, and the black will be the wild type. You see a predominance of red, which is what we were expecting, because I told you before that we knew beforehand that most of the good mutations in this type of system are transitions. So it's only normal that we see that the red one, which is good at producing transitions, it's generating a lot of mutations in this gene. But the big surprise came when we look at another loci, another locus on the chromosome, which is the PVP3 transpeptidase. Well, forget about the name. This is just the molecular target size of the antibiotic. Okay, and so we observed here a huge variety of changes. And I'm going to go slowly through this slide, but the idea is that I arranged the type of mutations that you have in these 3 different panels. Here you have all the changes whose underlying nucleotide substitution below is this transversion, okay, which happens to be the one which is very good in one of the mutators, in the blue mutator. In this panel here, I grouped all the changes that belong to transitions. Okay, so this is similar to what we discussed to the beta-lactamase, that we said that transitions were good for this enzyme. Well, here you have transitions, and they are not particularly good, but they are giving adaptation. Okay, and then in this panel you have all the other types of mutations, okay? And you see that this is enriched in black, which means that this is essentially what the wild type was doing. But the big take-home message from this panel is that when you look at the type of, you look at adaptation in this enzyme based on transversions, you see that most of it is blue, okay? So it's what you would expect based just on the mutational bias. And in this case, everything is red. I mean, if mutational biases wouldn't have much to say on the outcome of this process, you will see colors scramble around in this panel, okay? And so the idea, I mean, this is another way of looking at the data in which I'm arranging for each one of the mutators. I'm arranging the number of, so I'm looking at the alleles, either at the antibiotic resistant enzyme or at the molecular target, okay? And what you're seeing here is the combination of, having 1, 2, or 3 mutations in the enzyme, or having 1, 2, or 3 mutations in the chromosomal locus, okay? And you see that this is the sort of the transition prone mutator, and it's having sort of a mixture of both, although has a little bit more inclination towards substituting things on the 10 beta lactamase. You see in the enzyme, there is a certain number of triple mutants, which, by the way, are the ones that were also observed in Dan Van Rij's work. But the very interesting stuff comes when you look at the same type of analysis in the transversion prone mutator, right? Which you see a huge predominance of changes just on the chromosomal target site, okay? And so this is essentially telling us that those populations are not just favoring specific mutational changes at the nucleotide level that match their preference. I mean, that's something that, okay, it's not a big deal. But the big deal here is that they are preferentially exploring different mutational pathways that are involving different cellular target size, okay? And so sort of the conclusion from this work is that, yes, even in the high population size regime that we were exploring, cells were having over 10 to the 7 cells per milliliter. You see that just by including very strong mutational biases, you can happen to have populations preferentially substituting changes on one enzyme, or another population preferentially exploring changes in another enzyme, okay? So this is the first story that I wanted to share with you, okay? And this is just the story about that mutational biases. Well, I mean, in some cases, they really can matter on directing adaptation, okay? And so now I'm gonna move on to the second chapter of my talk. And I'm going to talk about something that is related, but completely different. I mean, some of you perhaps have started thinking that, well, if most of the mutations are matching just one specific type of nucleotide change, that could have some implication for, GC content of genomes, okay? So that was something that I had the opportunity to explore when I was doing my postdoc with Olivier Teneillon. And well, as most of you probably know, genomes in bacteria come in a great variety of sizes and compositions, from very large GC-rich genomes to very tiny and GC-poor genomes. And if you look actually to the whole distribution taken from GenBank, you see this sort of shape that people have been, trying to make sense of. In generally, the most widely accepted ideas now come along the lines of, you have strong mutational biases. I mean, this is something that it's already there. It was already there in the field. You have a deletion bias. Over the long run, genomes tend to get smaller, and just by what I mean is that, but just by mutational forces, and also you have a certain tendency towards substitute towards AT rich genomes. But of course, when you look at the whole distribution, you have a very wide range of things. And so people have been trying to put forward different, adaptive explanations as to what it's sort of counterbalancing these biases. So this is what I am depicting here with these arrows that represent selection, going towards, GC rich genomes. Perhaps because they are less biosynthetically costly. For instance, the selection in favor of increasing genome size is often invoked in case of generalist species that they need a larger repertoire. And so this is a very, very active field, and there's a lot of work going on. and it's really exciting as well. And so we wanted to check what would happen if on top of this picture, you also include the idea that, well, I mean, I already told you that mutators are something that happen in nature. What would happen if you have those mutators that are introducing an extra layer or biases, okay? And so, I mean, and we know from clinical settings and experimental evolution that mutators happen readily, but we also have evidence from comparative analysis of genomes that the DNA repair genes have been very frequently transferred among different species. Okay, so this very frequent horizontal gene transfer propensity of DNA repair genes is telling us that perhaps mutator episodes is something that happen in the natural history of lineages. Okay, so that's the sort of the background idea that we have from this work. And if that's correct, and we have some evidence that it might be correct, what will happen with this overall picture? And so this is all very nice, and we can discuss about that having a beer, but how do you go along and try to test this type of hypothesis in a meaningful way? So lucky for us, there's a very famous experiment in the field of experimental evolution. Most of you, if not all of you, already will know, which is called the Lenski's long-term evolution experiment. So Richard Lenski has been propagating 12 populations of E. coli in these little flasks with just a little bit of salts and glucose, a very, very simple environment, and has been doing this experiment since 1988. So it's more than 30 years, and regularly every 500 generations they have been storing samples from each flask, and they have all of that in freezers. And so there's an amazing amount of information that is coming up from this work. And luckily, there's something that could be useful for us. And that's because at different points in time, in this Lenski experiment, half of the lineages, that is 6 out of 12 of the lineages, became mutators, okay? And I shall bring your attention to the fact that in one of the lineages, mutation rate decreased at the very end of the experiment. Well, the experiment is ongoing, but very late. And we shall discuss about that later. For now, the idea is that you have this long-term evolution experiment, more than 30 years, and then you have half of the populations that at some point became strong mutators, okay? And so the idea was to look at what happened to the genomes of those mutators in Lenski setting, compare that to what happened in other type of experiments that people have already been publishing, which they do what they are called mutation accumulation experiments, which are basically propagating populations on bottlenecks of just one or very few individuals, so that natural selection cannot do anything, and what you recover is just the blind accumulation of mutations as they happen. Okay, these mutation accumulation experiments people have been doing lately with different mutators, and now they have been sequencing the genomes. So this information was publicly available, and that's a nice way of compare what will happen, what was happening in Lenski setting, in which that you have a very strong mutation rate, but for sure that adaptation was going on. You can compare now it with a case in which that adaptation is not going on, okay? And lately, as a third sort of approach, we can also look at the natural polymorphisms that you see between different variants, strains of E. coli, okay? And so the idea is that just by looking at what happened on those genomes, we can, perhaps learn something about the possible impact that those strong mutational biases can have on genome evolution. And so essentially what we did was we constructed this very big table with all the different changes that we observed, and we created, we divided on what we called selection-related factors and mutation-related factors. The selection-related ones are things that, are clearly obvious, like for instance, the effect, if it's a non-synonymous or a synonymous mutation. Also, we can calculate and score on what's the predicted disturbance that this amino acid change will create in the protein that happens to hit. And also you can look at the expression level, because we know that that's something that is interacting with the adaptive value of a change, whether the gene is essential or not, things like that. And when you look at mutation related factors, you just look at the genetic context in which the mutation happened. And so we just did our binary logistic regression, and I'm going to be looking at the odds ratios. So in another way to put that is on the different contribution of the different factors in observing changes, okay, in the genomes. And so here what you have is we're going to be looking at polymorphisms in natural strains, okay? And you see these lively colors on top of it are all those selection-related factors that I was discussing, okay, the synonymous versus non-synonymous, the effect on the structure of the protein, whatever, okay? And you see that in nature, these bars are very large. So this is telling us that selection is a major force in natural divergence. And you see all this tail on different shades of red, which are the what we call mutational related factors. And you see that mutational factors, which will be some time of biases that had to do with sequences, the sequence context. Those are also playing a role on shaping the genome, okay? But that's sort of our so to speak, our null case, okay? This is what happens in nature. Let's take a look to what happens with mutation accumulation experiments in which you are bottlenecking populations through just one individual, okay, serially. And as we expected, selection is, virtually absent in this type of on this type of experiments, and the only thing that you recover are, the effect of mutational biases, and so the question is, what will happen in the landscape setting in which you have mutators that are adapting to a well-controlled environment? And so you see something that it's in the middle, but it's not quite in the middle. I mean, if you look carefully, you see, of course, the signal of selection. And we know that because we know that those mutators have been, adapting at a very fast rate. And, over the 30 years of the experiment, the populations have increased in fitness over 60%, okay? So it's a huge amount of fitness gains. But still, the signature of selection is relatively minor. And you see that the dominant sort of signature are mutational biases, okay? So if you get the same data and you just look at the correlation, you see that the Lenski setting mutators, the correlation with what happens in nature is not super good, but it's really fitting like a glove with those experiments in which we know that selection is not going on, okay? So this is just telling us how mutators, even in very large population sizes that are actively adapting, they can, impose a toll on the genome, and they can really drive their GC content and composition, and also their size. And so just by extrapolating the magnitude of changes that we observed, it would only take 300 years, which is just 10 times more than what Richard Lesky had been doing. But as you know, in an evolutionary terms, in the macroevolution terms, that's the blink of an eye. It will just take 300 years to exert a change of 1% in GC content and to reduce genome size by 0.5 megabase pairs, okay? So the sort of the take-home message from this work is just that, well, I mean, as long as we know that mutators is something that are going to happen in the evolutionary history of extant lineages, those things are going to be blurring, are going to be really, making things a little bit more messy when we try to fit our theories based on selection versus deletion or AT bias to sort of explain the distribution of genome shape and composition. Okay, so this is the end of the second chapter of what I wanted to share with you. And so we are going to just proceed to the third one. Which is the idea that all those biases that we have seen that they can have something to say in driving adaptation. They can have some effect on genome GC content evolution, but also I'm going to show you that those biases can also be very, very important on the evolution of mutation rate itself. Okay. And so for that, I first have to give you a little bit of background on what we know about the evolution of mutation rate in asexuals, okay, especially in bacteria, okay. And so we have, I have to share with you the idea of mutator hitchhiking. And so this is going to be just a very simple conceptual model that I just want to make sure that you are with me on the same page, okay. And so the idea is that for any given, you know, asexual bacterial population that is growing in size until it reaches certain, I don't know, certain carrying capacity or whatever, you're going to have, if it's sufficiently large, you're going to have a fraction of mutators, okay? There is nothing special about mutators. You're going to have a fraction of a lot of different mutants, okay? Mutators happen to be very easily generated because most of them are loss of function mutations. And you see, I mean, it's very easy to simply destroy the function of a gene. So the fraction of mutators in natural populations, just by this sheer process of, you know, random knockouts, it's going to be considerable, okay? And so the idea is that you're going to have the subpopulation of mutators within a larger population of wild type cells. And at some point, you know, a hugely beneficial mutation or a bunch of beneficial mutations will create a selective sweep, okay? And, you know, everything will depend on the choice of parameters and everything and so on. But in general, given the fact that the wild type population is much larger, in most of the cases, it's going to be the wild type who's going to be substituting this this beneficial mutation, okay? And so what's going to happen is that the mutator subpopulation will become extinct, as will happen with many other subpopulations of, I don't know, knockouts in metabolic genes or many other things, okay? This is just another victim of the selective sweep. But of course, since mutators are very easily generated, as I told you before, it's just a matter of time until the next, you know, in the next generations, the mutator subpopulation will be replenished again, and you will be in the same scenario, okay? And so at the end, this is just a numbers game in which the idea is to see how often it's going to be the mutator subpopulation, the one who gets the beneficial mutation, okay? And that phenomenon that happens here, that you will expect that statistically will happen, you know, it's not going to be the most common, but the thing is that whenever it happens, you get the fixation of this mutator phenotype. So from this point onwards, the population will be, you know, 100% mutator, okay? And I told you that it's very easy to generate a mutator simply by knocking out one gene. It turns out it's not quite easy to revert one of these mutations. Generally, you will need this type of horizontal transfer events or something like that in order to recover the original mutation rate. And so this process in which the mutator subpopulation is just passively dragged along by a beneficial mutation is what is called hitchhiking. So it's this idea, you know, from how to stop that the mutator alleles are just hitching a ride with the beneficial mutations. Okay, so this is just the conceptual foundations of this story. Okay, and so of course you can do simple computer simulation models, and you know, depending on the choice of parameters, you will have different numbers. But the idea is that this is just the result from simulations in which you have, you know, time on the x-axis and then the frequency of cases in which one of the subpopulations fixes, okay, it finds the next adaptive mutation. And in this case, we will be watching at the wild type population, and you see that it's 90% of the cases for this particular choice of parameters. You see that in a very, very few percentage of cases is the mutator the one who gets clearly the upper hand. And then there's of course there's a mixed scenario, which I haven't shown in this type of cartoons, but you can imagine there is a mixed scenario in which both subpopulations will find, so to speak, the driver mutation. And then what will happen is that just one can fix. And generally, since mutators are generating not only beneficial mutations, but also, and especially, a lot of deleterious mutations, there's a lot of load associated to being a mutator. Whenever they both are in this type of competition, this sort of clonal interference regime, mutators will have the worst outcome. I mean, of course, we are talking in a case in which mutations are happening one by one, but this is just for the sake of clarity, okay? So in this work, what we wanted to see is to test to what extent this type of hitchhiking dynamics would be altered by the fact that different mutators have different mutational biases, and those biases can have different effects on fitness, okay? Because different mutators can have different access to beneficial alleles with different properties, okay? And so we essentially, we came across some of these pictures before. We essentially characterized the distribution of fitness effects for single-step resistance mutations for different mutators, okay, that have a contrasting mutational bias. And we observed those huge differences that I alluded to at the beginning. You see this mut-Y mutator, that is generating generally, not very good resistant mutants in streptomycin, but it's very good, it's very good in rifampicin compared to another mutator, okay, which is generating, predominantly smaller colonies in this antibiotic, but super big fat colonies in this other, okay? That will be what you see on the wild type, that is giving you a wider range of mutations. As you see also, there's more heterogeneity. And essentially what we did in this work was just to sort of estimate the fitness coefficients for these different types of mutations and use those to feed a computer model on the dynamics of hitchhiking. And so this is those three panels. We already went through them, and it will be the case in which you have no bias. Most of the cases is the wild type who is winning, okay? And because when you have also, this coexistence of beneficial mutations in both subpopulations, generally it tends to win due to the mutational load, the deleterious load. But something very interesting happens when you take into account bias, because it's not that much that things change in terms of the mutator getting things faster. I mean, this is not the case. What it's really going on here is that now, whenever we are in this scenario of coexistence, especially if you have, I mean, we are referring here to the case in which, I'm going to go back a little, in which you have, for instance, these very big fat colonies. This is the mutator with very big fat colonies, compared to the wild type that has a mixture, okay? And so statistically speaking, and overall, the mutator will be generating, better adaptive mutations. And so that can compensate all these deleterious costs. And in a lot of cases, when you have coexistence, the mutator will prevail. And I want to, bring your attention to the fact that this is creating a lot of impact, because we are going from just 0.6 success in this particular range of parameters to a case in which you go, all the way up to 12%, which is a massive increase on mutator success. Okay, actually, I'm not gonna go much into detail, but this is just a summary of the simulation. And this is the frequency of cases in which mutators sort of win. And in this axis, we have this mutational bias effect, okay? So that when it's 1, which corresponds to this vertical dashed line, that is sort of the classical case in which we are not considering any bias. Okay, so look at the triangles, the black triangles, that will be the frequency of cases in which mutator wins in this case here. But as soon as you allow mutators to have, on average, better or worse mutations, then you can have a huge change in mutator success. And mind you that this is a logarithmic scale. So this is telling you that just subtle changes on the mutational bias can change the hitchhiking dynamics by orders of magnitude, okay? So that's what regards the impact of mutational biases on the selection of high mutational variants of mutators, okay? But the last question that I'm going to share with you is what would happen in the other case in which you have a mutator population and it will evolve low mutation rates. That's something that, as I advanced before, we have one example in the Lansky experiment. This is one of the lineages that became mutator at some point. And in later generations, it evolved a reduced mutation rate, as you can see in this plot. This is the phase in which is a super high mutator. And this is in 40,000 generations, in which has reduced the mutation rate by almost 1 order of magnitude, okay? And so we, and I'm gonna be a little bit more quicker because I think we're running out of time. So essentially we used the data from the Lansky experiment to feed another computer simulation model in which we are considering, a perfectly adapted population of bacteria that is a hypermutator, in which an anti-mutator allele enters the population, okay? And we wanted to see how easily this anti-mutator mutation could reach fixation, okay? And of course, we again are playing with the idea that this anti-mutator mutation could shift the mutational bias so that on average will produce mutations that could be more or less deleterious, okay? And so you see, this is just the outcome from simulation. You see that just by changing the different propensity of the hypermutator and the anti-mutator to generate mutations with different fitness effects, you see a huge difference on the trajectory of fixation, okay? So we can extract selection coefficients from this type of trajectories, and we did a study that showed that for different, even for different scenarios in which your resident population is just a mild mutator that is increasing mutation rate 30-fold compared to the anti-mutator, all the way up to 1,000-fold hypermutator that will revert 100-fold when the anti-mutator enters the population. We see that this mutational bias effect again can have a very unprecedented and unanticipated effect on the dynamics of this system. And again, in some cases, especially when the anti-mutator tend to be the one that generates less bad deleterious mutations, we see orders of magnitude changes on the dynamics. And so to complete this sort of analysis, we wanted to look at natural genomes, okay, so real genomes, and just to make a very simple calculation, get all the proteins on those genomes, apply different substitution matrices to get an idea of the disruptive effect of different type of mutations, and see what happens in different genomes with different GC compositions, okay? And what I'm showing you here is two well-known bacteria, Staphylococcus aureus, that is a GC-poor bacteria, and Pseudomonas aeruginosa, which is GC-rich. And I'm showing you the average overall effect genome-wide of one type of transversions, which will be the red, and another type of transversions will be the blue, and then transitions, okay? And so you see that transitions generally are less disruptive. That was something that was already known. But we see a huge difference, reversing effect on transversions. If you extend this analysis to other genomes, you start to see, we are going here from GC poor to GC rich, and you see that those red and blue data points, they cross. We're going to see it more clearly in the summary plot with more lineages. And so this is what it's telling us, is that the actual effect of mutation, this is, of course, this is a statistical sort of reduction of the whole thing, but in an overall, on average, we will expect that the average effect of given mutational biases will depend on the GC content of the genomes, and that will change dramatically the type of dynamics that we were discussing before.
So,
but also,
Alejandro,
yeah.
we need to speed up a bit because we are running out of time. Okay.
Okay. So,
sorry.
Just the take-home message is that well, this has been a long journey just to tell you that, well, yes, of course, very strong mutational biases can affect directing adaptation. They can also play a role on shaping genome size and composition. But also, to make things even more complex, they have something to say about the evolution of mutation rate itself. And so that creates sort of a feedback complexity loop, because you can now think that, well, maybe genome evolution, depending on which type of GC content you have, it also will affect the propensity with which antimutators could change mutation rate, and so on. Okay, and so I just wanted to finish with this cartoon, which is again this idea of paths, and that, well, in this case is just depicting that in some cases when you start studying things, you end up finding out that explanations are more complex than what you expected. And just the very last slide is just the acknowledgement of the people that have been involved in this work and the founders that have been supporting me. So thank you very much for your attention, and I'll be happy to answer any questions.
Thanks a lot. Thank you very much. That's very stimulating, really. So I think we have already a couple of questions in the question and answer tag. So please do not hesitate to write them or to raise your hand if you want to ask a question. So I start with Roman Villutre. If you want to ask your question, please.
Do you hear me?
Yep.
Yes. Thank you for this very nice talk, which, as Ignacio was saying, is very inspiring. So when I saw your talk, I was, because I've read, but it's been a long time, about repeated evolution. And I was wondering, I mean, there has always been a debate of what is causing repeated evolution, but if mutational bias, if it should bias you towards, as you were saying, mutation towards the same gene, more preferentially, if it could explain actually what we seem to see in a lot of species, which is the repeated evolution of in some genes to adapt to the same factors in different species.
Yeah, well, of course, thank you very much. This is a hot topic, as you know, and because I mean it's not exactly what I mean the type of things what I'm doing, but people in the field of molecular evolution, they have recently realized that there is an important share of the convergent evolution that you see, when you sequence species, that can be traced back to preferential mutations, in particular hotspots in genes. Okay, and of course, I mean that's completely there's a clear link to the type of work that I've been doing. And what I would say is that I mean when you see a talk like that and you'll be working on that, at some point you have this impression that, well, but this is trivial. I mean, I should have known it, all the way along from the beginning. But from what I've been reading and listening to people that are working actually in the type of field you mentioned, on just looking at natural polymorphisms and try to see to what extent mutational biases are playing a role, there has been an historical sort of tendency to deny the effect of mutational biases, simply because the basic classic population genetic models, they were just concerned with showing the natural selection works, and we were in this in some cases in very simple scenarios in which you are thinking on very large population sizes, and as I mentioned in one of my slides, if you go to a very high, if you go to an infinite population size, it wouldn't really matter if you have local peaks. I mean, mutational biases will have no effect, and everything will be just as would you predict. You would predict just based on selection coefficients. Did I make myself clear?
Yes, totally. But as you mentioned, in a lot of species, the number of individuals is limited, or sometimes so the bias will be, I guess, stronger in that case.
Exactly, yes.
Well, thank you. That's answered my question. Thank you very much.
So then this the good there. So then now, Thomas Lerman, if you want to please to ask your question aloud, go ahead.
Hello. So yeah, you just mentioned a few points that I was about to talk about, because I think that this idea of mutational bias may refer to really different phenomena, and perhaps it would be nice to distinguish them. And in particular, perhaps you know, for instance, Richard Lenski wrote a paper in '93 about the controversy about directed mutation, and then there is which is slightly different idea, but related, and there is this idea that a bias, in mutational bias, could be just the fact that in some circumstances the average phenotype is changed on average by mutation. So that's really the definition of a bias. Mm-hmm. And then there is this idea that perhaps, you know, different mechanisms may have different mutational targets, and so may be more or less likely to occur by chance, because they just have different mutation rates. But that's mm-hmm. not, that's just more referring to the variance, mutational variance for different type of phenotypic change, and not really a bias. So, and in your talk, so it was kind of a mixture of all those definitions, and so I was curious to hear about what exactly you mean by mutational bias.
Okay. So I mean, let me think. How can I mean, this is a heavily conceptual question. Well, I mean, you first talk about this directed evolution, okay? And I think this is I agree that this is, some people could get confused by that, but it's not the case what we are talking here. Okay, so directed evolution, I mean, this goes back to the mutationist versus adaptionist debate in the early first half of the 20th century. And there were people that were thinking that perhaps mutation by itself could change, could direct adaptive change. Okay, and that was something that was completely erased from the modern synthesis. And although from time to time people come along and say, well, maybe there's some type of when people are talking about stress-induced mutagenesis, sometimes you have some echoes of this type of debate, but this is not the kind of mutation directed evolution that I'm talking about. I mean, in all my talk, and if I hadn't made myself clear, I will say now, I mean, the pushing forth is natural selection. It's just natural selection is the one that is causing populations to jump from one step from another. The thing is that just by presenting more often one of the different pathways, plus having certain limitation on population size, that it's not allowing all the pathways to be present at once, then you have a certain difference in the outcomes. And that's the type of mutation bias directed evolution that I refer to. So this is still natural selection, the one who's doing the job, but it's now that natural selection is getting overwhelmed by a particular type of pathway and say, well, okay, go along this way, as long as there is some adaptation going on. And then the third aspect you touched on was the idea of, correct me if I'm wrong and it's not what you're saying, but is what Paul Rainey it's been discussing over the last decade on genetic architecture. So the idea that by you have different phenotypic adaptations that could have the underlying structure that might be different, that could be involving completely different genes and pathways, and of course these different sort of modules will have completely different mutation rates. So just by the sheer fact that, in order to get a flagellum, you need, those many genes, the mutation rates will be this or that. And that also will channel, so to speak, the type of outcomes you see from repeated adaptation. Perhaps is that what you meant by in your third point?
Yes, clearly, but it's not only related to Paul Rainey's work. For instance, in your own data, when you mentioned that some paths are more likely than others, it seems to me that in many cases it's just that the mutation rate towards that particular path, that particular phenotypic change is higher, right? So which is not really a mutational bias in the sense, in the phenotypic sense.
I mean, you're completely right. If we go back to the example of the beta-lactamase, this enzyme that chews up antibiotic, and the chromosomal target gene, you just look at those two different sort of pathways. You see much more diversity of mutations within the chromosomal target site, which is just, again, referring to the idea that in this module, there's much more room for beneficial mutations. That just by that sheer fact, you will have more adaptation going on through this path. And I suppose as well, you are thinking on Evo-Devo, right? On the idea that, you know, in development, you even have a higher sort of structure of this architecture that we're talking about. There's much more modules that can be altering the overall propensity of a particular mutation to change one phenotype or another. I mean, this is, I think it's a fascinating stuff, and you know, I'm just trying to do little experiments with bacteria and little computer models and try to learn something, but you know, it's massive thing. I think because we are touching very fundamental aspects of evolution, right? I mean, mutation and selection. It's
okay. Thank you.
Good, thank you very much. I know. So, as a good Spaniard, they give you, so someone asks you a 10-second question, and then you give a 10-minute answer. We have the time for them for one more question, one more answer. Luis Miguel, probably you can go.
Yes, thanks for the talk. I was wondering if I understood well the last part of your talk. You were saying that mutators are more or less selected depending on the bias they have and whether it aligns with the direction of selection, more or less. So perhaps. Did you wonder how this will influence the evolution of the mutation bias itself, or the direction of mutations? To align with the direction of adaptation, because now you've shown in the experiments that different mutators, so you can think of this as modifiers that have different biases or different phenotypic directions, if you want, or molecular directions. So depending on whether selection is often aligned in a given direction, you will have more mutators of this type or that type that would be selected. It will change the genomic pattern of mutations, wouldn't it?
Well, I mean, I think that's you're completely right. I mean, this is a super exciting possibility, and I haven't given much thought, but I know that there is a paper on bioRxiv as we speak now by Deepa Agashe, and I think it's Lindy Wall as well, in which they are actually treating this very, very specific topic on the evolution of the bias itself. Okay, so I don't know very well the conclusion. I mean, they have a computer model, and they're showing that under certain conditions, it could really, you know, I mean, you can speak about the evolution of the mutation bias itself.
Okay. I will check it out. Yeah.
You will check it out. I mean, it's really, we are talking we're on the cutting edge of knowledge, you know, it's
yeah, no, but thanks that I've checked this out. All right, that's all.
Good, then thank you very much. It's really I think it's a field that I find personally very stimulating and exciting. Thank you very much, Alejandro. It has been a pleasure. So you will find this presentation in the YouTube channel in a few minutes. Please think about what Lorraine was telling us about the call for speakers that will be sent around in the next days. And looking forward to see you next in 1 week for sharing more science. Have a good weekend.